what we thought we'd do this morning is get a little bit more into some of the details of genetic association studies. And I think we forgot to mention yesterday that we did want to get some course evaluations from you, so we'll be passing those out at the break. And if you would fill them out at the end, obviously, that would be very helpful, and just leave them at the front there. So, okay. So the things that I was going to cover this hour were discrete and quantitative traits, measures of association, false positives, quality control, which is a big issue, then sort of initial looks at the data with what are called QQ plots, odds ratios, which you all are quite familiar with, but how they're calculated in genetic studies, and then a little bit at transmission and interactions. And because we're being quantitative, yes, I know that, Sydney, everybody knows that, but look, 4 wrongs squared minus 2 wrongs to the 4, yes, divided by 3 by this formula do make a right. So Gary was also a very quantitative person. Quantitative genetics is actually something that we don't tend to learn in high school and college. We tend to focus more on the qualitative components. But the quantitative is concerned with inheritance of differences between individuals that are of degree rather than of kind. I kind of like the way they put this, so I quoted Falconer and McKay. This textbook of quantitative genetics is really, it's very readable and really the classic in the field. So if you're interested in reading more, I would highly suggest it. So what are the differences? There are continuous gradations, obviously, rather than sharply demarcated types. The effects of the genes generally are small in order to give you a smooth distribution as opposed to, say, multiple means. Where in qualitative traits the effects are large, and then usually there are many genes in quantitative traits where there tend to be single genes inherited in Mendelian ratios for discrete traits. And I put a question mark there because actually, that was kind of the central dogma until a few years ago when it became very clear that there were important complex traits that were inherited in families in very sort of predictable ways that didn't seem to follow Mendelian ratios. So. Models for single gene traits say you have a big A allele that gives you a pink flower and a little a allele that gives you a white flower, and if you have 3 different genotype groups, if big A is dominant, anywhere that you have a big A, you're going to have a pink flower, as you see here. Anywhere that you have, if A is recessive, you need to have 2 copies of big A in order to have a pink flower. And if A is codominant, there are a variety of terms for this, but codominant is one that you tend to see fairly often. You'd get, with 2 copies, you'd get a really pink flower. With 1 copy, you'd get sort of a pinkish whitish flower, and then with no copies, you'd have your white flower. For quantitative traits, say that your big A allele gives you X units increase in height, and your little a allele gives you X units decrease in height. And say if these are of equal frequency, your population mean then would be some centered value 0, minus X and plus X would be the extreme values for your big A and little a alleles. So if you have 2 copies of little a and big A is completely dominant, both your big A homozygote and the heterozygote would be at this end of the spectrum. If A is only partially dominant, you wouldn't be right down here at the middle, but you would be sort of a little bit closer over to the big A homozygote. If A is not dominant or codominant, you'd be sort of smack in the middle. And you can even have, although it's rare, A being over-dominant if it's a heterozygote. And this may be some kinds of interactions between the 2 homozygotes or some other thing. Again, this doesn't happen very often, but it is wise to be aware that it could happen. The quantitative traits that to date have had published genome-wide studies. Actually, yesterday there was another one in protein levels, but for the time being, there are these that we can list. The Framingham study, as I mentioned, had these 18 groups of traits, many of which were quantitative, and I never quite know how to count them. Is it 16 or is it 34? But at least it's around 20-ish. And we went through an example yesterday in terms of how one looks at associations to get allelic odds ratios. So this, again, for a discrete trait, for myocardial infarction, 55% of your cases have the C allele, 47% of the controls have the C allele. This gives you a chi-square of 55 and an odds ratio of 10, and a p-value of 10 to the minus 13th. You can calculate what's called an allelic odds ratio, which is the odds of having disease if you carry 1 or 2 copies of the allele. So it doesn't matter how many you have, just having the presence of the allele. And here that would be 1.4. You can also calculate these by genotype group. Again, we did this yesterday. So the CC homozygote, 31% of the cases and 23% of the controls, while the GG homozygote, 20% of the cases, 28% of the controls. And again, a strong chi-square with 2 degrees of freedom now and a p-value at 10 to the minus 14th. Here you can calculate the heterozygote odds ratio specifically, which sort of lets these float free. It might be that the homozygote doesn't have all that much increased odds over the heterozygote. It'd be a little hard to explain biologically unless you truly have a dominant trait, which we tend to be thinking now may not be very common at all these days, at least not in complex diseases. But calculating a heterozygote odds ratio between these two groups is 1.5, and a homozygote between this group and that group would be 1.9. And this is the way that these data are displayed. And again, we talked about this yesterday with the log of the p-value here. And this is a nice example because they really found just sort of one very strong association on chromosome 9, 9p21. And this association has been found in many other studies as well. And mentioned that there are other ways of looking at these in addition. And here's another one, one for a continuous trait now, serum uric acid levels. What this group did was to regress inverse normalized levels. Why it was inverse normalized was just to make it transform into a normal distribution against the number of alleles. This was an additive model, so if you have one allele, you get sort of part of the effect, and 2 alleles, you get twice the effect, and they're sort of forced into that kind of a distribution. And even one can use covariates, and you can insert any covariates that you want in here. So it's nice to see that while with the qualitative traits, they tend to be relatively simple analyses just with chi-squares essentially, or even Fisher's exact test. With the quantitative traits, people are getting a little bit more sophisticated in terms of linear regression and adjusting for covariates. This one is another example in the uric acid literature, and showing here in 2 different cohorts. Here now are the mean uric acid levels in the 3 genotype groups, AA, AG, and GG, in these 2 different cohorts, and showing the additive effect for a single allele. Having one G allele drops your genotype mean, as you can see here in both of these cohorts. Association methods for quantitative traits that tend to be used, as I mentioned, linear regression, sometimes multivariable adjusted residuals, just to take out the noise effects of covariates. One can use linear regression of log transformed or centralized BMI. This was done in the Frayling paper. Variance components was very, very popular at one point in linkage analyses, and it sort of carried over into these studies as well. And so you can do a z-score analysis, as Sana did, at quantile normalized height, but there are, as I said, a variety of ways of doing this. Ways of dealing with multiple testing, and I think we talked about that a little bit yesterday. The family-wise error rate is sort of the general term that's given to corrections for either Bonferroni, the A prime, or the CDAC, which is a way of sort of correcting for the number of times that you subtract your p-value, your type 1 error from the universe of possible type 1 errors. I mentioned also the false discovery rate proportion of significant associations that are actually false positives, and the false positive report probability of Wacholder et al., which sounds very much like the false discovery rate, but is a little bit different. There's also a Bayes factor analysis. Bayesian analyses are sort of creeping into analysis of genome-wide studies, particularly with UK influence. So the Wellcome Trust did part of this in one of their papers. It is a challenge because you have to basically identify a reasonable alternative model that you're comparing your data to. So not a lot of people use it, but you may see more and more of it. So moving on to quality control, as these folks are doing, hold your horses, everyone. Let's let it run for a minute and see if it gets any colder. So unfortunately, with genotyping, we tend to sort of take it at face value, and there's sort of this in silico truth that you just assume that it must be right if it comes out of the machine. And there are actually a number of things that one needs to do to be sure that the data that you get are not artifactual. These include being sure that the samples that you have are the right samples and that they're high-quality samples. So we talked yesterday about how many satellites, variable number of tandem repeats, are used in forensic genotyping to identify individuals. And there are automated kits that are very easy to use and relatively inexpensive. Identifiler is one of them. I'm sorry, I don't know the company name or their location, but some of the genotyping labs will run these SNPs. These markers. They're highly polymorphic. They're, I don't know, 13 to 20 of them, and use that essentially as a barcode on a sample, so that whenever then you go to test that sample again, you run this barcode on it and make sure that you have the right sample, and it's a better label than anything you could stick on the outside of the tube. Blind duplicates, of course, make sense. What tend to be done rather than blind duplicates from within the study is duplicates of known specimens like the Ceph samples or any other standard samples from the HapMap. Gender checks, it's surprising how many women end up, in the prostate cancer samples and how many men end up in the female breast cancer samples, et cetera. This is a little bit more of a scary one because while you have a very good genotype measure, generally for sex, you don't have the best recording of that information, and it's sometimes incomplete or incorrect, surprisingly enough, and so those corrections can be made, but you wonder what other kinds of corrections needed to be made that can't be picked up genotypically. Cryptic relatedness, or what we sort of refer to as outbreaks of twinning, when there are duplicate samples in there that you didn't realize, and suddenly you have people who have exactly the same markers. They probably are not twins, but those are also things to be looked for, and we'll talk a little bit about that. Degradation of the samples or fragmentation is something that the laboratory should be testing for. The call rates greater than 80 to 90%. And we'll talk about those, heterozygosity and plate batch calling effects. So all of those are things that one looks at in the samples. And then once you've done the samples, you also want to look at the SNPs within samples. So again, you would look at duplicate concordance rates for a single SNP, because some SNPs are hard to type, just like some samples are hard to type. Mendelian errors, if you have family data, you can then see if there are errors in transmission. So you might have siblings where there are more than 4 alleles, which you shouldn't have, or certainly parent offspring where the offspring has things that the parents couldn't possibly have transmitted to them. Usually, only one of these will be accepted for a given SNP, and sometimes not even that many, but usually not more than that. Hardy-Weinberg errors, we talked a little bit yesterday about the expected proportions of the common allele homozygote, the heterozygote, and the variant allele homozygote, and those should be in binomial proportions if those conditions that I mentioned apply. So things like random mating, which actually doesn't apply, no in and out migration, no selection, and that. But in general, most SNPs tend to be in Hardy-Weinberg equilibrium, and usually the threshold for throwing out a SNP on violations of Hardy-Weinberg are fairly high. So 10 to the minus 7th, you have to have a pretty strong difference in distributions in order to question that SNP. Heterozygosity, and we'll talk a little bit about this. Based on population genetics, there should be a proportion of people who are heterozygous at a given SNP. It's usually in the 30% range. It seems to sort of fall there. A little bit higher for populations of recent African ancestry, a little bit lower for populations that are younger, as it were, or inbred populations where you wouldn't expect to have quite so many alleles floating in the population. Call rates for a given SNP. There are some SNPs that you just can't, just not sure from the intensity data that are generated what to call one of them, and so you leave one out, and I'll show you some examples of that. And you'd really like those call rates to be greater than 98%, and that tends to be the standard these days. It had been lower in the past, and a lot of those that were lower actually turned out to be artifact, and so we've sort of learned a lesson there. Minor allele frequency, which is the frequency in a population of the uncommon allele. And generally, alleles less than about a 1% frequency, there's some real difficulties in trying to genotype those. Now, this is going to be a challenge when we get to sequencing for even rarer and rarer SNPs that may be only 0.5% or 0.1% of the population. They may not be able to be typed on the existing platforms because they're not terribly sensitive to rare alleles. And then perhaps the most important thing is validating the most critical results on an independent genotyping platform. So you take the results from your Affymetrix array or whatever and then type them on something that's just specific to a couple of SNPs. And I mentioned Hardy-Weinberg yesterday, and again, here it is again. So the ideal conditions for it, and these are the proportions that you would expect to see. So these are some data from the Genetic Association Information Network testing the 2 platforms that we used on the HapMap data. The HapMap samples are available from Coriell repository. There's a small fee, I believe, for requesting them and sending them out. But they've been genotyped by everybody in the world, so the data are very stable. And what we show here is with the Perlogen platform of 481,000 SNPs, the Affymetrix platform of 439,000 that were used, and this is the 5.0 platform that were used in the GAIN study. These estimates of coverage are the number of SNPs in the genome estimated that are covered at 0.8 or greater, with an R-squared of 0.8 or greater. And this was really quite good for the European population, a little bit lower as expected for the Yoruba population. One can use just a single marker. Estimating is one marker in LD with another marker not on the platform. Or you can use information from a couple of markers nearby and use a multi-marker imputation, which gives you much more information using the LD that's surrounding that SNP. And there you can increase your coverage slightly. And the Affymetrix platform, sort of similar numbers, a little bit lower. The average call rate in both these platforms was over 98%, which is great. Homozygous genotypes, the concordance with the HapMap was 99.8% in the Perlegen for both, and a little bit higher for the homozygotes. Tends to be a little tougher to type the heterozygotes, and I'll show you why in a sec. Okay. And then some of the sample and QC metrics for the Affymetrix 5.0, which had about 500,000 markers, and the 6.0, which has about 1 million markers, just shown here. These are data again from GAIN. This was actually after we started doing the study. The previous ones were sort of gearing up for it in looking at the HapMap samples. And what you see here is that we threw out about 0.4% of total samples because they didn't pass a variety of QC metrics, and 0.55% didn't have a greater than 98% call rate. These numbers are obviously not exclusive, and we need to have sort of a flow chart of things as they kind of drop down through these metrics. With the 6.0, the percent failing was higher, but there are many more SNPs, and many of them are much tougher to type. So about 4% of the samples failing, and about 1.4% without a 98% call rate. And that's 98% for that sample across all of the SNPs that are typed. And then looking at the SNP by SNP quality, data quality information, again, about 500,000 and about 1 million showing here. We dropped out about 6% of samples that did not, 6% of SNPs that did not pass QC. Only 0.04% of the HapMap, of the 5.0 SNPs had a minor allele frequency of less than 1%, while in the much more dense platform, as you would expect, a larger proportion of them had less than 1% minor allele frequency. Greater than a 98% call rate, about 8% of the SNPs fell out on this metric, 9% on the 6.0 platform. If you drop that level to 95%, people tend to want to do this because they don't want to lose data, and there are many various combinations of babies and bathwaters talked about in discussing this. But at any rate, you'd lose about 4% in the 5.0 platform and about 4% here. The Hardy-Weinberg here now set at 10 to the minus 6, so really very stringent, and less than 1% dropped out for that. Mendel errors in this particular study with the 5.0 platform, we dropped out a lot because we had family samples. And in most studies, you don't have family samples, and you really can't tell. About the only way you can tell is if you have more than 2 SIBs, and you can actually compare, see that they have more than 4 alleles. So here we lost a lot, and less than or equal to 1 duplicate error, again, relatively small. So you can see here that it's the call rate that really tends to drop out your SNPs, and also the Mendelian error in that one particular study. This is a plot of heterozygosity. What one does is just basically, calculate the proportion of heterozygotes in the population for each SNP. And as you can see, it really kind of clusters around this 0.27 to 0.30 range. You can't see very well, but there actually are SNPs kind of shown along that kind of fall out here. And what I've done here is just to kind of blow up this area in the 100, the frequency of 100, and kind of dropped out those. So that now that you can see, there are some samples that are coming out here, and some out on this side as well. So we did have some outliers, and this tends to be done as basically right now. It's just you kind of look at it and say, those look a little bit odd, particularly way out here. Reasons for loss of heterozygosity tend to be, cancers can do it, but when you're looking at germline cells, decreased heterozygosity usually is due to genotyping error. Increased heterozygosity hadn't been worried about terribly in the past, and we learned a harsh lesson from GAIN that probably what this is telling us is sample mix contamination. So and it turned out that there were about 8 samples that actually had been contaminated with other samples in this particular gain study, and we didn't pick this up initially and then realized, hey, there's a real problem here. Went back and looked at these, and you can see that these are very small numbers of samples. In fact, there probably are about 8 of them here, and that was what the problem was. So all of these metrics, again, are evolving as experience accrues, and one of the things that we really want to do in GAIN is write up this experience and make it available to the scientific community. These are intensity plots for the genotyping data, what's calculated, and I don't understand the chemistry and I don't pretend to be able to explain it, but I know that what you basically produce for each one of the 500,000 or million SNPs that is typed, you produce a cluster plot like this, where you have, if there are 2 alleles of the SNP, you have allele A frequency plotted down here and allele B frequency plotted here. And as you might imagine, this would be the AA homozygote because there's no B frequency to sort of spread this out. This would be the BB homozygote because there's no A allele there. And then this guy is the heterozygote. And you can understand why it might be difficult to type the heterozygote, because you do have some bleeding over into this group. And these X's are things that are not typed. Basically, this is all automated. You can't sit and look at 500,000 of these. But basically, the calling algorithm, and there are a variety of calling algorithms, there are probably about 8 or 10 of them out there, but the companies, sort of settle on one that they use. And here the calling algorithm decided, I'm not going to call these, they're just too close on the border. These ellipses are kind of what the boundaries of what the algorithm will call, but as you can see, sometimes it doesn't call even something that's inside of an ellipse. So that's what these look like. This is a very nice looking one, although there aren't that many AA homozygotes, and this actually is the allele A is the T variant. And here, lots of homozygotes here, and nicely separated from the heterozygote group. Here is a little bit tougher one. There were no homozygotes, so this is a rare allele. So there are no AA homozygotes, but there is a heterozygote cluster, and there's a homozygote cluster for the common allele, and again, no calls. But here you've got much more of sort of a mess. So this is a badly clustering SNP, badly performing, and probably most labs would repeat this one and try to get it to the intensity to be much closer along the B line without anything in the A line intensity. And here you've got maybe these are the homozygotes and maybe these are the heterozygotes, but it's really very difficult to tell. What's recommended now, having gone through many of these studies, is that whenever you see an associated SNP, something that you want to call it 10 to the minus 7th or whatever level it might be, that you look at these plots and make absolutely sure that they look good, that you have 3 distinct clusters. So that means you don't have to look at all 500,000 or a million, but you do have to look at maybe 25 or 50. If you're carrying through 25,000 of them, that makes it a little bit tougher. And most people wait until they've gone through multiple stages before they start looking at cluster plots. And just one sort of kicker in this process, this is a paper published by Clayton et al. in 2005 that actually colored their SNP intensities by whether a sample came from a case or a control. So the red are cases, the blue are the controls. You can see the reds kind of clustering down here, and even difficult to see them there. And then this is just a different way it's a normalization of the intensity data. And you can see that there is a systematic difference between cases and controls. This could arise by different treatment of the case and control DNA samples. They tried very hard to control for that and make sure that the protocols were exactly the same. They were done in the same laboratories at the same time on the same plates and all of that. And they still ran into this. And basically concluded it probably had something to do with the collection at the site. Happily, this is something that can be adjusted for, so you can sort of calculate vectors associated with caseness and controlness, at least for a given SNP, and correct for it. But it is a cumbersome problem and one that needs to be looked for. Another thing that can introduce a systematic error is if you've amplified the samples in one group and not in the other, or amplified some of the samples and not others. Amplification is when you have a very, very, very small amount of DNA and you use polymerase chain reaction, PCR, to make as much of it as you like. You don't always get good copies of that, and it often gives you artifacts, and sometimes they look like this. So the laboratory that does this testing should be aware of these problems and should discuss them with you, but just so that you're aware of them. I wanted to get in a little bit into the problem of sort of unknown relateds or cryptic relatedness. Tom mentioned yesterday population stratification and looking at a population that's made up of a mixture of different groups. What's shown here is from the CGEM study, actually a little experiment that they did where they mixed together a whole bunch of groups of different geographic ancestry. And they started with the HapMap sample. So a lot of times when people do these kinds of analyses, they start with 3 population groups or samples that are known to sort of cluster tightly and to distribute out differently from each other. So this is the Yoruba from the HapMap, the Ceph population, and then the Asian populations. And they also typed an African American group, and as one might expect, they sort of fell on the mid-range between, and this is this principal component analysis, between the Yoruba and the Ceph population. They also typed a Native American and Latino group who tend to have more Asian ancestry alleles, or at least to cluster more with Asian ancestry groups. And you can see where the Native Americans actually clustered quite tightly here, Latino a little bit spread out a little bit more. And then they did a third principal component. So the way, and it's a shame that Kang isn't here because he could explain this much better than I can, but anyway, what one does is to look for vectors that are basically separating the samples in the most effective ways, essentially. And you can do this up to hundreds of principal components, but after the first several, you tend to run out of steam. So it sounds like we have some competition here. I hope you can all still hear me. But. Okay, so in doing this then, you notice that the Ceph population for the third principal component actually moved in quite a bit. So they got kind of pushed in. The Asians and the Yoruba stayed pretty much the same. The Yoruba shifted over a little bit. And here you're African Americans where you'd expect them, sort of between these two groups. And now the Latinos tend to sort of take off in this direction between the Asians. And when you go on to the fourth and fifth principal components, you have this tremendous sort of clustering of samples that you really can't separate out, and then you have a few down here that seem to be driving this, but it's not clear, and they tried going to a fifth principal component and still didn't much, and they're kind of wondering, what the heck is going on. So in looking at this group, they looked very carefully, particularly at these samples that seem to be outliers and that will be driving the vector calculation. And what they found actually was that many of these people were related. So these were 2 parent-child groups, and then there were half-sibs here, and these were half-sibs all along through here, who were all basically unsuspected in this sample. When they then randomly chose one person from each of the related groups, they were able to separate out their clusters very nicely. So this was kind of a surprise to many of us. This was presented at one of the last GAIN meetings by Gilles Thomas. And it's something that you need to be aware of, that related people do end up in your studies, even if you don't expect them to, and you may not always pick them up. What they pointed out was the studies that they were comparing, this again from the CGEMs, they had multiple studies around the world, the ACS, the PLCO, this is a European study and another Northern European and another European study. And you wouldn't think that there would be people in common across those studies, but as it turned out, certainly in the U.S. studies, there was a SIB pair that was participating in both the Harvard professional study, the health professional study, and the ACS. There were 7 people that we call socially conscious people who were participating in both studies. There were SIB pairs in the PLCO and 3 people in PLCO and the health professionals. So social consciousness is a risk factor for unexpected twinning in your data, and then SIB pairs here and a father-son pair here. So this does happen. It's probably in your studies. If you're not aware of it, you will find it with genotyping data. And if you go looking for a structured population, when you have 2 people that are that closely related, it really sort of points the vector at it. And it can also do other things in terms of clustering when you're trying to cluster your SNPs and your data that really can mess up your analysis. So kind of summary points for genotyping quality control. Sample checks are done for identity, for gender error, for cryptic relatedness. Sample handling differences can usually be adjusted for, but you have to be aware of them. And doing an association analysis is often the quickest way to find your genotyping errors. You'd like to think that that's how you find your real signals, but it's not. One of the things that we tried to do in GAIN was actually to make all of the data, the association data, the results, available to everybody at the same time. We wanted GAIN to be, as forthright and sort of public community resource as possible. And in doing that, we basically said to the principal investigators and the genotyping labs, we're not going to tell you who's a case and who's a control or what SNPs are associated with what. We'll just show you the data kind of blinded, as it were, or masked to the SNP identity, and it made it very, very difficult for us to be able to sort of, separate out these things, and certainly doing quality control and figure out who was related and who wasn't. And we decided that blinding was really not the best way to go about this, and so we now have a sort of a back and forth with the investigators who provided the samples, and we allow a very short time for that, because obviously quality control can go on for months and months, but it is a challenge. And rare SNPs are the most difficult to call with these kinds of platforms, and just be aware of that, particularly as we get into rarer and rarer SNPs as we do more and more sequencing. And inspection of genotyping cluster plots really is crucial. Okay, once you have your data cleaned up, what tends to be done is to look at the association statistics, and taking an easy example, this again from the Easton study, you basically take your chi-square statistics, you plot them along, there's an expected distribution, I didn't label this, sorry, so this is expected chi-square, and remember, if you have no association whatsoever and you just keep choosing randomly samples, you'll end up with the chi-square distribution, the normal distribution squared. And this is the observed chi-square. So if your observed population basically follows the chi-square distribution, that is, there's no association, you would expect all of your plots, because you're just lining up your chi-squares, all of them to fall along this little gray line here, which is the identity line. If they don't fall on that line, it means that they're not following that distribution for some reason, which could be genotyping artifact, or it could be that there's a real association there that's sort of pulling them upward. And what you see in this black line here is that indeed there are some departures from the chi-square distribution, and some of them are actually, like this one here, relatively dramatic from what would be expected in the chi-square distribution. These red dots are corrections for the population structure in this population. So what tends to happen is if you have differences in ancestry between cases and controls, they will inflate these statistics. You can correct for that. And one way of correcting for it is this, what's called a genomic control, or lambda value. We won't go into this in detail, but you just basically divide all of your statistics by whatever the lambda control value is, and that was done here, and that's what you end up seeing. So this one actually for Easton didn't look all that dramatic, but then when they started doing the replications of their top SNPs, it did sort of pop out some results. You'll also see people publish tables like this, where they just basically plot out a table of the observed, then adjusted for that genomic control, and the expected values by the significance level, so 0.01 to 0.05. You'd expect to see 934 based on the chi-square distribution, and they observed 1,239, corrected down to this level. So, a modest excess of modestly significant, or really not very significant at all, SNPs. And then going down to the increasing levels of significance until they got to 10 to the minus fifth, and they had 13 times as many. They would have expected to see 1, and they saw 13, which suggested that they actually did have some important associations outside of chance. You tend to see these tables much less commonly. People like to look at pictures, and so you tend to see QQ plots much more commonly. This is a very nice one from Hafler et al. Looking at multiple sclerosis, and you can see the identity line here is this red line. This gray line is their initial associations, so their expected distributions, and then what they observed, and you notice this dramatic takeoff. Multiple sclerosis has long been known to be associated with the MHC, multiple histocompatibility complex, major histocompatibility complex alleles, which are very, very diverse and are very heavily genotyped on these platforms. So you have lots and lots of SNPs. It's actually not all that many, but it looks like a lot, that are sort of taking off for very high chi-square values. If you then take out the major histocompatibility complex SNPs and just plot what's left, you end up with this here, and you're still having some strong departures from the expected distribution. So you need to be aware that sometimes people will take out a particular locus, or a locus that's already known to be associated, and kind of see what's left. This was done for prostate cancer in the Icelandic study. Again, they show them at different scales. So here's your identity level. The blue is corrected. The red is uncorrected. You have very strong associations shown here. But knowing that chromosome 8, that AQ24 region we mentioned yesterday, is known to be strongly associated with prostate cancer, they basically took out all of chromosome 8 and then looked again, and still you have a departure from the expected level even after you correct for, most of these kind of fall out once you correct, but you still have a few SNPs that are outside the expected distribution. And this is the QQ plot for myocardial infarction that I showed you yesterday. I think I showed it to you yesterday, but it's from the Samani study in the New England Journal that was part of the Wellcome Trust Case Control Consortium. And here you can see there's quite a strong distribution from the number of that are departing from the expected distribution. And these actually are all of the SNPs that were had a p-value of less than 10 to the minus 7th, which they sort of declared as their initial threshold. And when one looks at the actual plot now arrayed along the chromosome, those 30-some SNPs are these here. So this is a nice plot because they're all sort of, hanging right on top of a particular chromosome. And one can then sort of look at not only this level of significance, but this group is also departing from what you would expect in the expected distribution. And that is basically corresponding to this level of association. So these were SNPs that the Wellcome Trust group said, they're not decisive in our study, but they are sort of interesting. And as we explained yesterday, with the SNP number 24,000 in the initial association ending up being one of the top SNPs in a replication study, they said, we certainly would want to look at these as SNPs of interest, as it were, in a subsequent study. And then there's this area here that has a modest departure from the expected distribution, and that is probably all of this here. And what those are most likely are SNPs related to structure of that population, so differences in ancestry that probably are just noise, but one needs to be aware of and adjust for them. And then this is the plot from that study. These are the 39 SNPs that they found to be strongly associated on chromosome 9. And here's the one that I mentioned yesterday, 3049, known by its nickname. So this is the most strongly associated SNP, and this is just another study that they did, a replication study. And here again is that what I showed you yesterday, the Boston to Providence sort of map distances. And you can see that here you've got one linkage block, so maybe these SNPs are independent of the associations with these because you do have a recombination hotspot here. And these are all sort of clues that you can get as to where the kind of operative or important SNP might be. Something that I think Tom mentioned at one point yesterday was the winner's curse, which is the tendency for initial studies to overestimate the effect, the magnitude of an association. And this happens in all kinds of studies. It was described most colorfully, I think, in genetic studies. And it's shown here for what was probably the very, very first genome-wide association study. It was done with only 20,000 SNPs, so we tend not to count it in our lists of studies, but it was actually Ozaki et al. In a Japanese study of myocardial infarction looking at lymphotoxin A, and you'll see in their study they found a stronger odds ratio than any of the others found, 1.71, quite significant. And then in subsequent replication studies, these all sort of fell back toward essentially no association. When a meta-analysis was done of all these studies, it was significant. It was not significant, 0.98, and probably this was a spurious association that looked good because when one does lots of these, you end up picking the extremes, and those are the things that get published. So again, a little publication bias, a little winner's curse. Getting into very briefly gene-gene interaction kinds of studies. This is a genome-wide scan looking at Alzheimer's disease, 861 cases, 600 controls. This is a little bit hard to see, but you probably can pick out this one red line going up here. This is on chromosome 19, and it's the ApoE locus. And it's very, very, very strongly associated with Alzheimer's disease. This was known before the study was done. It had been identified in linkage studies, as we talked about yesterday. And as you can see, the p-value here is probably about 10 to the minus 40th. So very, very strong association. But is there something else in here or isn't there? What was done by this group then was to, was to then stratify and look just at the people who were carrying ApoE4, so sort of get rid of the impact of ApoE4. They actually stratified people with it and without, and now, of course, vastly expanded the y-axis. But you can see that there are a couple that do sort of pop out as being associated with, particularly here, with Alzheimer's disease in the E4 carriers, suggesting that there might be some interaction, and these were not associated in the E4 non-carriers, and the sizes of the groups were, reasonable enough that you would expect power was not an issue in the other group. So they then looked at the odds ratio of late onset Alzheimer's disease associated with this particular SNP 3115 in the GG homozygotes by their E4 status, and found that people who did not carry any of the E4 alleles had no no association with Alzheimer's disease with the SNP, and those who were carriers of the E4 allele had about almost a threefold increased risk if they also carried this particular, if they also were homozygous. These are small numbers, and they don't give you the numbers, but you can see the confidence interval is relatively generous. And in looking at everyone together, there still was an association of this particular SNP. So all of that sort of coming together to suggest that there may be some interactions between this particular variant, which is GAB2. I'm not familiar with it, and it's being looked into by the Ryman group in terms of what that interaction might be. This is a genome-wide scan for age-related macular degeneration. I think I showed this yesterday. It was probably the March 2005, earliest truly genome-wide scan at 100,000 or so. They found actually 2 of their SNPs that went above their genome-wide cutoff level. Turned out that this one was genotyping error, and it went away when they looked more carefully at the genotype. So 1 out of 2 ending up being genotyping error. One can calculate population attributable risks based on odds ratios and prevalences from these studies. That was done here. And you can see that here the association. These are 2 SNPs that were sort of right next to each other. You couldn't tell that on the plot. But they sort of calculated these for both of them. Here's the more strongly associated SNP. The frequency was quite high, and the odds ratio for a dominant model was also quite high. So population attributable risk, which is essentially a function of the prevalence and the odds ratio, was very high for the homozygote and even for the heterozygote. For the heterozygote, the homozygote odds ratio was higher, but obviously the prevalence was lower, so the attributable risk was lower. So the feeling was that this may even be almost a single gene disease. These odds ratios have come down somewhat in replication studies, so it now looks like it's about 2 or so, but it still accounts probably for a large amount of this disease. What was interesting about this variant was that it was then taken forward into the nurses' health study, and various environmental factors that might be interacting with this very strong risk factor were examined. And these are data, Schomburg et al. Published these. These are data that are available in any cohort study and that could be looked at for any of these SNPs, if they've typed the SNPs, and is work that is begging to be done and really needs to be looked into. So what they did was just to stratify based on obesity, less than 30 or greater than 30 BMI. And use the group that had this is actually, I believe this is the variant allele that's protective, but it's the ancestral allele is the one that's a risk allele. But at any rate, this group being the comparison group, and you can see that as you carry more copies of the H allele, your risk goes up whether you're lean or obese, but your risk goes up more if you're obese. And similarly, if you're a non-smoker, you have increased risk. If you're a smoker, you get considerably more increased risk based on both smoking and on the prevalence carriage of this allele. So there's an interaction here. Neither of these were quite statistically significant. They didn't reach a 0.05 level, but they were certainly suggestive. And again, this is work that is not going to be done by geneticists. It is work that could be done by epidemiologists. And while at times when we raise, we sort of raise the specter of, don't you think you should be looking for gene-environment interactions? Very often the answer we get back is, yes, but there's multiple comparison problems. And you think about it, you say, wait a minute, you did, 1 million SNPs. I'm only doing 5 environmental factors, or 1,000 environmental factors. It's still not going to be nearly as bad. So that's work that you should just be aware it needs to be done and could readily be done. One of the nicest examples that I've seen of gene-environment interaction has to do with some work that Jose Ordovas did on the hepatic lipase C or LipC genotype in relationship to HDL. And basically, these are smooth plots as you might expect. And what he showed was a relationship between fat intake and HDL that varied by the genotype at this particular locus. And so basically, in the TT genotype, the more fat you eat, the lower your HDL. In the CC genotype, the more fat you eat, the higher your HDL. Now, these are ecologic data. They're not interventional data. So forgive me for implying causality, but that was what was inferred essentially. What's interesting about this is, say you looked at this middle band at people in a population who were eating about 30% fat, which is about the average American diet, you would conclude that there was no association between LipC genotype and HDL level, if that was the question that you were asking. Whereas if you looked at a population with very low fat intake, such as from developing nations and that, you would conclude that the TT genotype has high HDLs, the CC has low HDLs, and that this is codominant with the CT being in the middle. If you were then to look at a population with a very high fat intake, such as Scandinavia, you'd conclude that no, it's not the TT that's associated with HDL, it's the CC, and it's actually a dominant effect, because you can see it here in the heterozygote, and then the variant, the TT has the lowest HDL levels. So this is how you can, tend to miss these kinds of associations or get inconsistent relationships between genotypes and phenotypes, and we need to be aware that this happens all the time and is rarely looked for. So gene-environment interactions can be an explanation for inconsistency in associations. And I did come across another example of this. This was endotoxin exposure and allergic sensitization by CD14 genotype. And again, if you looked at the relationship of this genotype to probability of sensitization to this particular allergen, you'd conclude that there was no relationship with genotype. If you had a sort of a middle of the range endotoxin load, if you had a low range, you conclude one thing about the CC homozygote, and if you had a very high range, you conclude the opposite about the CC. So gene-environment interaction can be quite important, and is something we should be pursuing. So I think I'll close by just saying there are challenges in studying gene-environment interactions, as you might imagine. The genes actually are pretty easy to measure. That's why we can measure so many of them at such low cost. The environment is very, very difficult to measure, and hopefully it's getting easier with more automated measures, but it's something that the Environmental Health Sciences Institute is investing a fair amount of money in, and I know a lot of work has been done here on that as well. Variability over time, it's hard to argue that the DNA sequence varies over time. Whether it's turned on or turned off probably does vary with time, otherwise we would all keep growing and we'd keep developing and that. But for the most part, the sequence has low to none variability. The environment obviously can often have high variability. Whoops. And recall bias, there's probably none in the genes. It's certainly possible in the environment. Temporal relationship to disease, as I mentioned yesterday, is pretty easy for the genes. It's kind of hard for the environment. And just to close with being aware of one's environment, I guess I'll have the ham and eggs to the surprise of the chickens and pork here. So thanks, and I'll be happy to answer any questions. I have to say I was delighted to see that it was sort of rainy and gray today, because I felt so guilty about keeping you all in on a beautiful sunny day yesterday. So, yes?
When you're talking about sample handling differences, where along the chain are you talking about? Anywhere?
Really anywhere. And some of that may be, it may have to do with the participants themselves. So samples can be different from young people versus old people, just in the way, your DNA might be isolated in that, certainly in the transformability of it and all. But really any step along the way. And I think there is investigation going on in many of the laboratories now, trying to figure out what are the steps that, what are the things that really perturb it a lot. But granted that we can't really figure out what those steps are, at least look for it and try to adjust for it if you can, if there are systematic differences.
And then what about, you mentioned genotyping errors. Is that on the platform itself, or?
It's probably calling error more than on the platform. It's basically that the chemistry didn't work well enough to be able to separate out the intensity of the two alleles. Now, there are other things that could cause genotyping error. What if you have 3 alleles? That would be one. Or if you have a null allele, you're not going to have any intensity for that person. It's not clear how to count that. If it's deleted, basically, so you don't have that SNP. Or if you have copy number variants, you may have 4 or 5 or 6 copies of it, and where does that show up in your intensity? And there are algorithms being attempted to sort of come up with those, but most of it is genotyping error and clustering error. Okay.
Kind of look at the principal component analysis. One of the slide, few slides you showed there.
Do you want to go back?
Yeah.
Okay, I'm really bad at explaining principal components, but I'll do my best. Sorry,
fine.
don't look at the screen if you tend to have photic seizures.
Close.
So this one, or before that?
Previous, yeah. First
Okay.
and second,
First
third,
and
and
second.
fourth.
Okay, I'll make these
Okay,
guys
that's fine.
forward. Okay.
So what kind of variable you were used to construct this principal component? What kind of?
This is allele frequency. So
Allele
it would be
frequency.
frequency of, and what I believe the principal components does is pick out the SNPs that have the most differences between groups.
Or just pick a few allele or all the allele?
I believe it picks, whichever alleles are the most different between groups. And it could be one, but it's probably many of them.
Okay, all right, thank you.
Sure. Yeah, and the nice thing about principal components is because they're linked, they're in LD, you would get a bunch of them would sort of come down with one component.
Yeah, I guess they pick up several earlier because here there's several principal components, so it is.
Oh, there are many, yeah. Yeah, I mean, it can go out to, 100 principal components and more, and then you start, separating out sort of clans and towns and things like that.
Okay, thank you.
Sure.
You just mentioned that the gene-environmental interaction can be an explanation for the different results in the different studies. And of course, through yesterday and today, over and over it was mentioned that we need replication studies. So how do you know then if we know that several cohorts do not collect the same environmental factors? So one example, for example, in the nurses' health study showed a positive, strong association, and nobody else shows that this association was real there. So how do you account for these differences? Because then you cannot make a statement, yes, this is a false association, therefore there's not.
Yeah. No, it's a huge problem. I mean, obviously we can't make all of the cohort studies collect the same information. You'd like to be able to at least have some comparability between them or collect some kind of watered down version, and the problem, of course, is then it's watered down, and so are you missing a lot of the variability within there. I think what tends to happen when there are inconsistencies is people just dismiss them, and that I personally think is a mistake. There may be some very important information there. The problem is, when you have a million possibilities, you want to at least separate down, distill down the ones that you really think are really interesting or potentially important and try to pursue those. But you have to be, selective.
Right, and this goes back to then wouldn't be necessary, and I know that it's already out, the idea of having this huge cohort that your institute was thinking about. Therefore, because of these issues, wouldn't be necessary for the future to have this huge cohort?
Yeah, we think it's really necessary. We don't see how we're going to get to sort of the bottom of this without a very large cohort that is well phenotyped. The large cohorts that are going on now are not that well phenotyped, and the environmental characteristics that are collected are even poorer. But yeah, we think it's necessary. So.